We have developed a novel technology, antibody membrane switch, which is a novel way to display antibody on cell surface. And this membrane anchorage of antibody is actually switchable, so you can remove it from the chromosome after you isolate your desired cells. So the purpose of this technology actually is to facilitate fax-based high throughput screening of the cell lines. So one of the immediate applications, it could be used for cell line development. You can isolate the cells with a high expression of membrane antibody and then remove the membrane anchorage and transform the cells into production cell lines. We're also in the process of applying library screening with this technology. So our ultimate goal actually is to streamline the antibody engineering or antibody discovery and the cell line development into a one-step, 3-month process. I think one of the trend in this industry is improved product quality. I think antibody as a biologics, large glycan proteins, has been always thought to be a mixture of, because of the different level of glycosylation and different level of post-translation modification. So you don't actually have a very precise molecular nature of your product, even for the commercial antibody therapeutics. I think one of the trend of the industry is to how to improve the quality and really define the critical quality attributes. Yeah, I've been to the conference a couple of times. I think it's a great place to meet, to see all the new technology, to see all the progress of a variety of front of the antibody engineering, and particularly for the therapeutics. I think to reduce the cost is a key issue. I think as a society, you just cannot support 100 to 200K per year of therapeutics. I think as a society, we need to reduce the cost. And on the other hand, we need to enhance the quality and the drug quality, and particularly to really define the product quality attributes, and then you don't always give a patient a mixture of your drug, which maybe some percentage of drug are not really beneficial or might even causing immunogenicity. So you really want to define your nature of your drug at a molecular level. Particularly in my field, how to enhance or how to improve cell line development, I think one of the key questions is how to access the product quality attributes in the early stage of cell line development. And I think that would greatly benefit your quality control of the final product. There's always many opportunities, I think, in terms of antibody therapeutic development. And what I can see the opportunity is how to reduce the development time from a target. Well, after you validate a target, how to get to a efficacious antibody therapeutics in a short amount of time. So I think conventionally we spend probably 2 to 3 years for the antibody engineering and to really isolate a drug candidate, and then another at least 1 to 2 years for cell line development and maybe process development. I think one of our goals is to really streamline these two processes into a one-step and 3-month process.